Welcome to Neutralizing COVID, the monoclonal antibody story, the podcast where we unpack the science, strategy, and real world impact of monoclonal antibodies in the fight against COVID 19. I'm your host, Dr. Shikha Agrawal, a biochemistry and molecular biology researcher with a passion for demystifying the science behind life saving treatments. In this series, we'll explore how these precision engineered molecules took on SARS-CoV-2, dissecting their science, clinical milestones, triumphs, and setbacks. Across all the episodes, we'll dive deep into how these precision therapies tackled SARS-CoV-2, spotlight a few key treatments, explore their challenges, and look ahead to what's next for antibody therapies in respiratory infections. Whether you are a clinician, researcher, student, or science enthusiast, this podcast is your comprehensive guide to one of the most compelling chapters in pandemic therapeutics. To kick off this series, we are diving into the immune system's first line of defense. Welcome to our very first episode, Unmasking Our Microscopic Defenders, How Antibodies Fight Back. To help us unravel this complex topic, we have a special guest with us today, a PhD in pharmaceutics specializing in nanomedicine and cancer drug delivery, Dr. Aiswarya Chaudhuri. Welcome to the show.
Thanks for having me, Dr. Shikha. It's a pleasure to be here and discuss this exciting area of science.
So let's start with the basics. We hear the term antibody a lot, especially in the context of fighting of illnesses. Could you explain what antibodies are and what they do in our bodies?
Absolutely. Now, antibodies are the proteins that our immune system produces in response to foreign invaders like bacteria, viruses, and other pathogens. Now, you can think of them as highly specific security guards. Their primary job is to identify and neutralize these threats. Each antibody is designed to recognize a very specific part of a pathogen called an antigen.
So it's like a lock and key mechanism.
Precisely. The antibody, which is the key, has a unique structure that allows it to bind very tightly to a specific antigen, which we can call the lock, on a surface of the invader. Now, this binding can have several effects. It can directly neutralize the pathogen, for instance, by blocking its ability to enter our cells, or it can flag the pathogen for destruction by other components of the immune system.
That's a great explanation. Now, we're specifically talking about monoclonal antibodies today. What makes them different from antibodies our bodies naturally produce when we get sick?
Okay, when our body encounters a virus, say the flu virus, our immune system mounts a response by producing a wide array of different antibodies that target different parts of that virus. Now, this is called the polyclonal response, where poly means many. Now, monoclonal antibodies, on the other hand, are a collection of identical antibodies. Here, mono means single. They are all clones of a single parent antibody cell, and therefore they all recognize and bind to the exact same spot, the same epitope on a specific antigen.
So instead of a broad attack, it's a very targeted, precise strike. How are these monoclonal antibodies made? It sounds quite high tech.
It is. The process typically starts by identifying an antibody that has a desired effect. For example, one that is particularly good for neutralizing a specific virus. This might come from a patient who has recovered from an infection, or it can be generated in the lab. Now, once that specific antibody producing cell is isolated, it fused with a type of immortal laboratory cell line to create what's called a hybridoma. Now, this hybridoma can be grown indefinitely in culture, producing large quantities of one specific or monoclonal antibody. Now, there are other methods too, like recombinant DNA technology, which allow for even more controlled production and engineering.
Appreciate the deep dive there. Building on that, how about the structure? What does a typical antibody look like, and how does that structure enable it to do its job so effectively, especially against viruses?
Now, antibodies are typically a Y-shaped proteins. Now, this Y has a few important parts. And the two arms of this Y-shaped antibody are called the Fab regions, which stands for fragment antigen binding. This is the part which that recognizes and binds to the antigen on the virus. Now, the stem of the Y is called the Fc region or the fragment crystallizable. This part interacts with other components of the immune system, like certain immune cells, and can trigger them to destroy the virus once the antibody is bound to it.
So the arms grab onto the virus and the stem calls for backup.
In essence, yes. Now, for viral neutralization, the Fab regions are critical. They can, for example, bind to proteins on the surface of the virus that the virus uses to enter our cells. If these sites are blocked by the antibodies, the virus simply can't infect the cell. Think of it like putting a cover over the keyhole, so the virus can't unlock the door to our cells.
That makes sense. Now you mentioned engineering. Why do we need to engineer these antibodies? Are the natural ones not good enough?
Uh, Natural antibodies are fantastic, but there are some reasons for engineering them. Now, one major reason, especially when antibodies are first developed in non-human systems like mice, is to make them more human-like. Now, if we introduce a purely mouse antibody into a human, our immune system might recognize it as a foreign body and attack it, reducing its effectiveness or causing side effects. So, we use genetic engineering techniques to replace parts of the mouse antibody with the human sequences. This leads to chimeric or humanized antibodies that are better tolerated.
So it's about reducing side effects and improving. How will they work in humans?
Exactly. Now, engineering also allows us to fine tune other properties. We can, for instance, modify the Fc region, that is the stem of the Y, to enhance its ability to recruit other immune cells, making the antibody more potent at clearing the virus. We can also engineer antibodies to have a longer half life in the body, meaning they last longer and might require less frequent administration. Now, for viruses that mutate quickly, like influenza or SARS-CoV-2, we can also try to design antibodies that target regions of the viruses that are less likely to change, making them more broadly effective against different variants.
This brings us to COVID-19. Monoclonal antibodies have been a significant part of the conversation in treating or preventing severe illness. How do they specifically work against the SARS-CoV-2 virus?
Now, most of the monoclonal antibodies developed for the COVID-19 target the spike proteins of the SARS-CoV-2 virus. Now, the spike protein is what the virus uses to attach to or enter the human cells, binding to a receptor of our cells called the ACE2. And the monoclonal antibodies are designed to bind to the specific parts of these spike protein, particularly the region that interact with the ACE2 receptor. Now, binding to the spike protein, these antibodies can physically block the virus from attaching to our cells, effectively neutralizing it. So, some can also prevent the virus from fusing with the cell membrane, another crucial step for the infection.
Very insightful, Dr. Aishwarya. So they essentially form a shield around the virus's key, preventing it from unlocking our cells.
That's a very good analogy. And because they are so specific, they can be very effective. The key is to administer them early in the course of infection, before the virus has had any chance to replicate extensively and cause widespread damage. They have been particularly explored for individuals at high risk of progressing to severe COVID-19.
That was a great overview. Let's continue by exploring the emergence of different viruses of SARS-CoV-2. How does antibody engineering help address these evolving viral threats?
That's a really good question and an important one too. Now, the viral evolution is constant a challenge. As a virus mutates, the parts of it that antibodies recognize, that is the epitope, can also change. So if an epitope changes significantly, the monoclonal antibody that was previously very effective might lose its ability to bind and neutralize the virus. This is where the ongoing research and antibody engineering are crucial. Now, scientists are constantly working to identify new antibodies that can neutralize the emerging variants. One strategy is to develop cocktails, which means two or more monoclonal antibodies that bind to different epitopes of the virus. Now, this makes it much harder for the virus to mutate in a way that escapes all the antibodies simultaneously. Now, another approach is to engineer a bispecific or a multispecific antibodies that can bind to two or more different targets on the virus, or even a target on a virus and a human immune cell simultaneously to enhance the immune response.
It sounds like a constant race against the virus, Dr. Aishwarya. What does the future look like for monoclonal antibody therapies, not just for COVID-19, but for other viral infections as well?
The future is very promising. Now, the technology for discovering, developing, and manufacturing means monoclonal antibodies, are advanced tremendously. We are seeing them explore for a wide range of viral diseases like influenza, RSV, HIV, Ebola, and many others. There are also a lot of research into making them more accessible and easier to administer. Now, for example, through a subcutaneous injection instead of intravenous infusions. Furthermore, the ability to rapidly respond to new viral threats, as we saw with the COVID-19, by quickly developing targeted antibody therapies, is a significant step forward in pandemic preparedness. We are learning how to make them more potent, long lasting, and broader in their activity against viral variants.
It's truly remarkable how science is enabling us to fight these microscopic battles. Before we wrap up, I just want to remind our listeners that while we have discussed the science behind monoclonal antibodies and their use, this podcast does not provide medical advice. Any decisions about treatment should always be made in consultation with a qualified healthcare professional. Dr. Aishwarya, this has been an incredibly insightful conversation. Thank you so much for sharing your expertise and making such a complex topic understandable.
It's my pleasure, Dr. Shikha. Happy to contribute.
A big thank you to our listeners for tuning in to Neutralizing COVID, the monoclonal antibody story. If this episode sparked your curiosity, don't forget to subscribe, share this episode, and join us next time for more on monoclonal antibodies in COVID-19. Until next time, take care and keep exploring science.